Thank you very much to the organizers, especially Chen Yuzhang and Dr. Qian Chen for the invitation. So I'm very, very happy to share our recent progresses in the language model to facilitate understanding of cellular signaling. So my name is Yu Xu. I come from Huazhong University of Science and Technology and Hubei Hongshan Laboratory at the Wuhan, a beautiful city in the middle of China. So if you have any questions, please feel free to interrupt me at any time if you want. So we know that cellular signaling is very, very important, lies at the heart of all biology. So actually, cellular signaling is also the fundamental language of the life for the cells to communicate, to make decisions, and to coordinate cellular functions. So we know that actually the dysfunction of cellular signaling is one of the important core hallmarks of many human diseases, such, for example, the cancer. So you can find that in cancer, signaling, many circuits were organized to the module to be dysfunctional. And for the cellular signaling, this process can be broken down into 3 steps. So the first, the signal will be perceived usually by the ligand receptor interactions. The receptor usually locate on the surface of the cells. Then the signal will be transmitted into the cells through a number of cascade intracellular events, mainly depend on the post-translational modification, for example, phosphorylation, ubiquitination, and acylations, and so on. Then the cell will make a response, make a decision on the signal. For example, to induce the change of transcription regulation of some specific genes. So understanding the cellular signaling is really, really difficult. So how to understand cellular signaling? Actually, we know that biological discoveries usually have 2 paradigms, namely hypothesis-driven and data-driven. So today, I will use an example to describe the importance of these 2 paradigms for the discovery of the self-immune tolerance, that why our immune system does not attack ourselves. So we all know that the Sakaguchi have a hypothesis. The hypothesis is how is the immune self-tolerance achieved. So based on this hypothesis, they designed a set of experiments, and finally they found a subpopulation cells of the CD4 and the CD4 positive and the CD25 positive cells. They named these cells as the track cells, regulatory T cells. They found if this subtype, the cells, is absent, so the immune system will attack the own tissues. But however, when these cells will be added, so the self-tolerance will be achieved. So this is the typical hypothesis-driven paradigm. However, the functionality of the track cells is not known. It's quite difficult. So this is the problem of the hypothesis-driven. Usually, these studies will open the doors, but actually the underlying mechanism is difficult to be understood. So in parallel, so we also know that the data-driven paradigm is very important. For example, the studies from the Branco and Rumsdale, they studied on the scurf mice. These mice will have very serious autoimmune disease. So they used a data-heavy method named random shotgun sequencing without any predetermined target. So they just sequence, and they finally found a 2 base pair insertion will be causally associated with this disease. However, you know that, so this is also the importance of data-driven without any hypothesis, but the new finding can be identified. So 2 years later, Sakaguchi will combine the findings from the 2 paradigms, including the hypothesis-driven paradigm, including the data-driven paradigm, together, and they finally found that the FOXP is the key regulator of the track cells. So we all know that both paradigm is very, very important. So for the hypothesis-driven paradigm, actually the efficiency is very low. So we all know that this mainly dependent on human thinking, so it's very low. but actually its interpretability is very high. So on the contrary, the data-driven, the efficiency is very high. However, currently its interpretability is not very high. So each paradigm have its superiority. So we all know for the data-driven paradigm, we all know that AI empowered augmented. The first example, actually, the success of AlphaFold. So if you know how to play the Go, you will find that when the Lisado, sorry, whether I can use this, yes. So when the Lisado put the white piece at here, if for human players, you will put the black piece here to protect the middle, or put the piece here to protect the border. However, you know that AlphaGo put the piece here. So this step cannot be imagined by human. So that means machine is not just memorize. It learns the rules underlying the Go place. And also the same example is the AlphaFold. So yet night, Angela told us the story or published AlphaFold on Nature, right? So AlphaFold actually is the language model to learn the grammar of protein language on the structure and on the sequence. So this deep learning-based method is really important for the data-driven paradigm. So now we know that in biomedics we have many foundation models. These models cover the genomics, cover the transcriptomics, proteomics, drug discovery, and single-cell analysis. And these models are starting to facilitate our understanding of the cellular signaling. In my own lab, so my main research interest is focused on the post-translational modification. And we also are interested in how to use the AI technology to analyze cellular signaling. So during the past 2 decades, we developed many tools including 19 predictors, 5 tools, and 14 databases. So these resources have been integrated at the platform of Akuku. So first, that actually we did a lot on the AI-ready database. So we know that the data actually the fuel of any model, deep learning model. So we collected 2.8 million phosphorylation sites, so much larger than other databases, for example, Phosphosite Plus and other databases. We also have the largest data set on the lysine modification sites, about 600,000 lysine sites. And also we pay attention on the PTM-related cellular processes, for example, autophagy, that is the self-eating process, and also the cell cycle circading, and so on. So based on this data set, I will give you an example, concrete example, how the data-driven paradigm can help us to understand the biology. So the first example is the GPS-6. So this example is use the general phosphorylation size, about 500,000 phosphorylation size, to pre-train a model to learn the general grammar of phosphorylation. Then we use a much smaller and specific kinase with known kinase associations to fine-tune the models. So this pre-training followed by fine-tuning, the paradigm is very useful to handling on the limited data set. So how the results? So you can find that GPS-6 can predict PK kinase specific P size for about 44,000 kinases in many species. And by comparison, you can traditional methods such as Mix Plover and the Deep Zero, they can only predict 100 kinases. So our coverage at least 100-fold increase than other tools. And this tool is very easy to use. So you can find that you can put the sequence here, one or multiple, sorry. Whether I can use it? Oh, sorry. So you can put the sequence in the text form, and you select the kinase, and then you click on submit. So the results will be shown, including the kinase, the peptide, and the score, and also the web logo, and so on. So it's very, very convenient for following experiments. So predicting phosphorylation size or PTM size is one thing. However, understanding its function is another thing. So prediction functional relevance of the PTM size is very, very difficult because we know that only a very small proportion of PTM size have been reported to be functional. For example, in our database, only about 1% PTM size have been reported to be functional. Current analysis also indicates that actually a very important PTM size likely to locate on the important structure place on the protein, for example, at the active sites, at the disorder region, and so on. So 5 years ago, Pedro Bartra group will develop a tool named FunScore. So they compared a large data set because we know 2,000 functional P size is a very large data set. So they combined many, many features and use the traditional machine learning method. They develop accurate predictors to predict the functional relevance of PTM size. However, for many PTM, you know that the reported size is very, very small. For example, we studied a newly identified PTM named lysine BHB size. So for this PTM, only nine sites have been reported to be functional. So we all know that for the traditional machine learning, you cannot handle such a small data set. How to do it? So I'm very thankful to my intelligent student, Xinghe Huang. He is a girls student. She developed a very, very useful method named PFunk. So PFunk, the framework PFunk is very, very similar to nesting door. So we first used about 100,000 lysine modification sites to pre-train a model to learn the general grammar of the lysine modification. And then this model will be fine-tuned to the KBHB to reinforce the grammar of the KBHB. And then we use the 9 sites to further fine-tune the model. So we use this hierarchical framework. We can use the very, very small, tiny small data set to accurately predict the functional relevance of PTM size. So we also learn to interpret the model. So you can find that we predict the accuracy for the predicted lysine PTM size is accurate. For the KBHB size prediction, it's accurate. And for the functional KBHB size, it's also accurate. And we also analyze the features contribute for the prediction. And it's very interesting. You found that when we predict whether a site will be modified or not, so we call it as the specificity. So the important feature is actually the in-contact feature, named the radial interactions not very near, maybe in the distance. So when we use when we fine-tune the functional model, you will find that structural features is more important. So this is also aligned with the intuition of human biologists that the PTM functional important PTM sites prefer to locate in the important position. So we know that new predictions always bring new findings in biology. So we indeed screen our results and we focused on a very important protein named aldolase B. Aldolase B has been studied by Dr. Shen Cailin in the Xiada University. He identified this protein as a glucose sensor. So in the high glucose, this protein will bind to the IBP and locate on the F-actin. So it's not free. So when the low glucose, so this protein will release its substrate IBP, and then locate on the lysosome to inhibit the mTOR signaling. So this signaling is very important for the cell growth. So we use the pFunc to predict the two sites on this protein. But actually, you know that the algorithm cannot tell which site is really important. So we did. So my collaborator named Da Jia in the Sichuan University, he led a team to do very great experiments. So they found that use the ketogenic diet consumption, it really increased the aldolase KBHB levels in both two sites. However, we found that mutating only the one site is important. So mutating this site will inhibit the IBP binding affinity for about 50%. And it also inhibits mTOR activity and block its lysosome localization. And eventually, such a modification will suppress tumor growth. That means ketogenic diet can reshape the cancer metabolism through the KBHB. This is the first case on the KBHB function in physiology. So besides KBHB, we also ask another question, whether this method can be extensible to other PTMs. So you can find we have lysine acetylation. So lysine acetylation will have over 500 sites. The method is okay. And we also validate our predictions. And you found two PTMs, including the chromatination. They have only 21 sites. And the lactylation only have 11 sites. So both data sets can be efficiently dealt with the pFunc framework. That means this can be a general framework for the functional PTM discovery. So let's have a summary. The takeaways of this section is that actually we know that the data-driven paradigm actually is augmented by the learning-based artificial intelligence methods. So on the probe side, so this method can accurately pinpoint the key candidates. It's generate highly valuable information for the experimental design. On the con side, so the accuracy need to be improved because data is really, really small. And also, we all know actually the interpretability need to be improved because this method usually generates a probabilistic score, maybe from 0 to 1. So how to interpret such score is very, very difficult. So we ask another question. So whether we can also augment the paradigm of hypothesis-driven. We all know that actually the emergence of the general purpose large language provide the potential for this paradigm. So also the Dr. Gao Xin also tell us the importance of ChatGPT and the DeepSeek and so on. So you know that for this language models, many emergent capabilities. For example, chain of thought. So chain of thought, that means the machine can break down a complicated problem step by step to resolve it. Very similar to a human. So that means we will have a shift from a pure machine learning that only depend on the probabilistic predictions to the machine reasoning. Use more structured and human-like common sense to interpret the results, to interpret the causality. So this is very important to augment the hypothesis generation. In the last year, so we published a paper in the Nature Communications. So I think this is the first attempt to test whether the ChatGPT can be assistant in science for to analyze biology. So our promote is very simple. So we want to know how many genes are associated with the three behaviors, including sleep, sociality, and locomotor. So we pilot you to interpret, for example, our promoter is that, can you tell me the function of this protein and whether this protein is involved in regulating sleep? So we compared a large data set with the known genes involved in three behaviors, not used for training, just used for testing. So you found the performance actually is not promising. So you can find that actually the specificity is okay, but the sensitivity is about only 20%. So that means the AI hallucination is strong. So how to reduce the AI hallucination and to improve the high quality response? So we have one paper accepted named Lanmao. So Lanmao have the pronunciation very similar to the Lanmao, blue cat in China. So this character is a very famous cartoon character in China many years ago. So we all know that massive omics data is very important to identify cellular signaling networks and require mechanistic interpretation. So in this framework, we combine the GPT, ChatGPT for the biological knowledge reasoning. And we also use the deep learning to predict the context-specific predictors in different contexts. So we use the autophagy as an example, and we interpret a large data set, and we expand the knowledge of autophagy regulator, context regulator. And very interestingly, we identified a receptor for the COVID entry. Is very important for autophagy. And we also prioritize the new therapy for the cancer. So I will tell this story. So autophagy actually is a very important signaling pathway. It will involve to form a half cup brain to engulf many cytoplasmic contents and then seal with the ball to deliver the cellular contents to the lysosome for degradation. So beside the core machinery, many context regulators can involved in the autophagy to have different functional context. However, we know very few on the context-specific regulators. So in these studies, I'm very thankful to my collaborator, Professor Di Peng, and my postdoc, Da Chao Tang. They help us to develop a workflow. So the left part is the reasoning part, and the right part actually is deep learning part. So we combine the reasoning and the deep learning together to identify new regulators. First, actually, how to use the ChatGPT. We all know the left is the simple. It's casually ask whether this protein is involved in a cellular process. No. So this promoting is not so efficient. So we use to need a loop or chain of promoting, including the background, question, and confidence. So you can first ask what the function of this protein, and you next ask whether this protein is involved in some cellular signaling, and you need to add. So please use the 0 to 9 to indicate the possibility. So use this one, you can find the hallucination will be suppressed to generate high quality results. So use this method, we can identify molecular of interest from the literature, not new, but known. The next we also know that molecular will be functional usually to interact with other proteins. So protein form to the network. So if we want to know the molecular association, we need to use the chain of thought reasoning. So this also the chain of the promoting is the background. So maybe you need to, can you tell me the function of this protein and this protein? This is the background. And then you will ask the question, so whether this protein, you will tell them that this protein are involved in autophagy because it's derived from our experiments. And whether this protein have relation to each other. So based on your available biological knowledge, can you tell me the chain of thought? So you request the ChatGPT to answer in the chain of thought manner. So this will generate a very high quality response. For example, EML1 is the newly identified autophagy regulator in our study. So you can find that use this promoting the zero-shot promoting the AUC actually is promising, not very high, but it's okay. In the human, in the mouse, in the rat, and in the yeast. So use the ChatGPT, it will interpret about 10% of the genes will be associated with autophagy, but in general, not in the context. And AUC values is promising. So we all know that from the knowledge, you can only get the general knowledge. So you cannot get the context-specific information. So if you want to predict the context-specific predictors, you need to use the real data, the omics data. So we compared a lot of omics data from the literature, covered many conditions, many types, including transcriptomics, proteomics, and phosphoproteomics. So we first construct the protein-protein interaction graph. Then we use the omics data and the interaction features to train this model. And we use this model to predict the context-specific regulators. So you found that the performance is promising, very promising. And also it expands the knowledge of the autophagy. And we validate indeed, because my lab also do experiments. So we validate our prediction by ourselves. So you can find we identify new autophagy. That indicates LIMOI can reveal real biology. So based on these results, we also reason its underlying mechanism and finally identified actually ATG1 is the downstream effect of these two genes, two related in the autophagy. So then we started a last studied autophagy named DSF. So DSF is an alcohol abuse drug, but it also has anti-tumor activity. The mechanism is less understood. So we use the omics and LIMOI to predict the two proteins and the screen together with the screen. So we found two proteins are really important in the DSF-induced autophagy, and the silencing either will reduce this autophagy process. So we also reasoned the underlying mechanism of these two genes. So you can find that many autophagy core machinery genes will be regulated by two proteins. And interestingly, LIMOI interpreted these two proteins are both anchor proteins. So we do experiments, and we found that indeed the autophagy pathway is necessary for the cancer cell survival in our context. So we also use the tissue, cancer tissue, cervical cancer tissue, to find that the two protein expression is negatively correlated with the patient survival. So we also use the mouse model to validate our results. Then we know that we do not need AI at all. We just use the human is okay, because we know the autophagy is important. Autophagy is important for the cancer survival. So whether we can use this autophagy inducer, including a CTSL inhibitor, for combination therapy, it's indeed you can find that when the two drugs are added, the tumor will be potently inhibited. So that means this is the typical reasoning augmented discovery from a drug with mechanism unknown to identify specific regulators and lead to new combination therapy. So I also summarize on this section. So we for the reasoning augmented hypothesis-driven paradigm, it has many pros. It also has many cons. So our recent study is whether we can integrate the two paradigms to further augment understanding of cellular signaling. So I will quickly on this study. So we developed a framework named 荀子, and we used this one to identify new kinases in the Parkinson's disease and cancers. And also this is the 荀子 architecture. So the left part is the hypothesis-driven, and the right part is the data-driven. So we use the hypothesis-driven and the data-driven for the enhanced biological knowledge discovery. So for the take-home message for this study, we all know that cellular signaling is highly complex. So it determines the health and the disease, and actually it's the language highly dependent on the context. So we currently know that AI empowers actually both paradigms. For the learning augmented data-driven, we have GPS, and it can help us to find needles in haystack. And also reasoning augmented hypothesis-driven, we also have two like Moy, and it will tell what those needles do. So also these language models facilitate our understanding of the cellular signaling and to do new findings and so the future plan. So we hope to develop a hybrid system. So finally, thank you to many of my collaborators and friends, including Chen Yuzhang and Jia Dang. And also, I need to thank my students, their creativity and the hard work behind every slide that I show you today. So thank you. So thank you very much. Thank you. So please ask any question. I will be very happy to answer it. Thank you.
Questions?
Thank you, Professor. Very impressive work. So the fact that LIMOI can find some new regulator for autophagy is really impressive, and I was curious about, so how do you think that, how difficult for a student or a well-trained PhD student to do the same hypothesis, or is something that can't be, is really counterintuitive? Yeah.
Thank you. It's a very great question. So, okay. So actually, my background is cell biology. So my graduate students in a cell biology laboratory. So I understand the logic of biology, and for training students, actually, I have a idea is that sending our student to the wet lab to train. So for simple experiments, I think we need to do by ourselves to learn more on the biology. But for very complicated biological experiments, we try to collaborate with others. So we cannot do very complicated experiments. So this is the one thing. Another one thing for the machine learning or for the biomedics, actually, I think this I cannot teach students because you know that actually I'm not very so young, and actually the artificial intelligence is quite difficult to me. So students learn by themselves. I just ask them to discuss with them. So thank you very much. Thank you.
Any other question?
Professor Xue, in one of your previous method, I saw that a method about the context-aware regulator prediction. I saw the PPI knowledge graph is served as a source for that. So may I ask how that work?
Actually, a very good question. Our thinking is that if we just use the transformer to learn the relation between two genes, such information may be not reliable. So we can use the topology of protein-protein interaction networks as a predetermined information to filter not necessary information. So this will help us to fasten the training process and to prioritize really important or real results. So this is our solution. Thank you very much. Thank you.
Thank you, Professor Xue.
Thank you.
Okay, our next speaker will be Professor Robert Andrews, who is a professor of systems biology from Imperial College, United Kingdom. He will talk about his work on AI.